Welcome everybody. Today we're starting a course on the application of bacteriophages in biotechnology. Well, first of all, what is biotechnology? Biotechnology is a field of science that uses biological systems or living organisms or parts of organisms to create something new, to create some new product, to develop this product. So this is biotechnology. And bacteriophages are living creatures that can be also used in biotechnology. But first, before we start the conversation about the application of bacteriophages, I would like to tell you just few words on the biology of phages, because it is important to know the subject, to use it well, to get more results of it. So our subject is bacteriophage. Bacteriophages are viruses. These viruses infect only bacteria. They are absolutely safe for humans, animals, and plants. They can only kill bacterial cells. And the name bacteriophages means that they are bacteria eaters. They eat bacteria, they destroy bacteria. First, the bacteriophages, or as they are referred now, phages, were discovered in the beginnings of the 20th century. Almost simultaneously by two scientists, Frederick Tward and Felix Durrell. And actually, Felix Durrell gave this name bacteriophages to this group of viruses. Well, phages, like viruses, they all have their protein capsid that contains genetic material. But phages are a little bit more complicated in their structure. You can see that this is the structure of a common bacteriophage. They are also called tailed because they have tails. So phages have capsid, or it is called head. This head contains genetic material. For most phages that are used in biotechnology that are known, this genome is presented with DNA molecule. Sometimes it can be RNA molecule. Well, anyway, for most phages, this is DNA. So, head, genome. Also, phages have tails, and this tail has an inner core. We will talk about this core a little bit later. Also, very important part, base plate with tail fibers and pins, and this part is used by phage to connect to bacterial cell. So, this is the common structure of a bacteriophage. Like all the viruses, phages can be seen only under the electron microscope. You cannot see a virus or a phage under the light microscope, only electron. And you can see the picture of a phage, and it refers to the scheme that I already gave you, the head, the tail, and the tail fibers. Well, phages can also differ in their structure. Some of them are spherical, some of them are filamentous, like rods. But all these phages can be used in biotechnology also. But the most common structure still is phage with head and tail. But tails can also be different. Some phages have tails that can contract, contractile tails. They contract to inject the phage's DNA into the cell, like a syringe. Some phages have tails long or short. They do not contract. But these phages, all these phages, can use a very interesting enzyme, endolysin. This enzyme melts the cell wall. It can also kill bacteria, and this enzyme can be also a subject of interest for biotechnology. So, what is also very important about all the viruses, especially phages used in biotechnology, is also their structure and the structure of their capsid, because it is built of the protein subunits, repeated protein subunits. What does it mean? It means that this is one and the same protein in many copies. So we can either change this protein for our protein of interest, and we can get big amounts of protein of interest with the help of phages. Also, we can add the gene that codes our protein of interest to the genome of the phage, and it will express the new protein on its surface. So we can also get a sort of system with new proteins that can be used, for example, in the creation of biosensors. So, when the phage connects to the cell wall of the bacteria, as I already told you, it contracts its tail And its inner core, remember we talked about it, the inner core breaks through the cell wall and the phage injects its DNA into the cell. So its genome gets into the cell. But the phage itself remains outside the cell. So this capsid protein doesn't get into the cell, only the genome of the phage. And here what happens next. When the phage enters the cell, it has two variants to develop inside the cell. Some phages are called virulent or lytic phages, and they cause lysis of the cell. They kill the cell. How does it happen? When the phage DNA gets into the cell, it starts the mechanism of destroying the cell genome, and only the genome of the phage remains within the cell. And the cell, the bacteria, becomes the factory to produce phage proteins and phage genome. When there are enough materials already created, phage particles start to assemble. And when those particles are absolutely ready, the cell bursts and the virus, the new generation of bacteriophages, comes out. So this is the lytic cycle. But not all the phages cause lytic cycle. Most of them are temperate. They can cause either this lytic cycle or lysogenic cycle, lysogeny. It means that when the phage enters the cell, it doesn't kill it. It stays within the cell, either like molecule, it can be called plasmid, or it enters the genome of the cell and becomes a prophage. It just sits quietly within the cell genome. It doesn't reproduce. When the cell divides, each daughter cell receives its copy of the phage, the prophage. And this is the passive reproduction of a phage. It remains within the cell. It doesn't reproduce like this. So now we come to a very interesting question. First of all, why do we need to know about this lysogeny? Because temperate phages cannot be used to cure bacterial diseases, because they won't kill bacteria. They will stay within these bacteria. By the way, very interesting question is that those tiny creatures, those phages, only a protein capsid with genome, they can actually make a decision how to act. They really make a decision whether to choose the lytic cycle or lysogenic cycle. So how do they make this decision? Well, simply, like for example, human beings do it. If we want to build something, we have the materials, the desire, the energy to build something, and we start the building. The same thing happens with the phage. When it enters the cell and the cell is rich and there is a lot of energy and materials to build new generation of phages, so the lytic cycle starts. But if we don't have enough materials, desire, energy to build something, we will just sit quietly and wait until the desire comes or the moment comes. And that's what happens with the phage. When it enters the cell, and this cell is poor, it grows in a poor medium, it doesn't have enough energy and food to build new particles, so the phage prefers to sit still in this cell and wait until the good times come, or the bad times come. We will discuss it. So, when the decision is made and the state of lysogeny has started, we must remember about lysogenic conversion. This is the state of a cell which has a prophage. When the prophage stays within the cell, it can bring new genes to this cell. And these genes can change the abilities, the qualities, the characteristics of this bacteria. For example, if we have a pathogenic bacteria that received also a prophage, it can become resistant to antibiotics. It means that this bacteria, without the phage, was sensitive to antibiotics, and when this bacteria received the phage, it became resistant to antibiotics, cannot be cured with antibiotics. So the state of lysogeny, lysogenic conversion, is very important if we talk about phage therapy. And also, the induction. What is it? So when the phage entered the genome of the cell and it sits there, it can also be pushed out. When the cell is stressed, for example, impacted by some physical or chemical factors like ultraviolet radiation, X-rays, or some poisons. So the cell is stressed, and it starts the mechanism of checking itself, whether everything is all right. And also it checks its genome. Special enzymes check the genome, and if they find the prophage, they are kind of like, uh-oh, this is something wrong, and they cut it out. And the prophage is inducted. This process is called induction. So this prophage becomes simply a phage that starts a lytic cycle. So lysogeny can be stopped by induction, and then the lytic cycle, lysis, starts. So this is the inducted phage, and you can see that new phage particles are created and they break through the cell. So this is lysis of the cell. This is what happens. Okay, so now lysis and lysogeny both are parts of one and the same life cycle of the phage within the cell. The phage enters the cell, enters the genome, becomes prophage. When the cell divides, the prophage divides with the cell, and this is the passive reproduction. If the induction happens, the phage starts the active reproduction, and in the end, the bacterium is dead, and the new generation of phages is released outside. Now, we've discussed the basics of phage biology, only the basics, and we can go on with the application of bacteriophages in biotechnology. First, and very interesting, antibacterial therapy. Well, when the phages were first discovered, they were actually planned to be used as antibacterial agents to kill bacteria, to fight bacterial diseases. But this therapy was unstable, the results were unreliable, and shortly after the discovery of phages, the antibiotics were found. When the antibiotics were found, they gave stable results, they were killing all the bacteria, and phage therapy was put on the shelf. Today, it receives the second breath, the second life, because multi-resistant bacteria appear. They are called superbacteria, superbugs. They are unsensitive to all the known antibiotics. So a person who is sick with the infectious disease caused by these superbugs cannot be cured by any antibiotic. I'm not saying that all the humanity now is facing this problem, but individuals are already facing this problem, and the only aspect that can help them is the biological one. The natural predators of bacteria, the phages, they can kill bacteria. So, antibacterial therapy. Also, phages can be used to detect bacteria and to type them, because the interactions between bacteria and phages is very specific. Specific to the species, and we can sort of classify bacteria with the help of phages. The next application, vaccines and drugs delivery. If you heard already about DNA or RNA vaccines, so if phages are used as vectors to deliver these vaccines to the organism, to the microorganism, so the results are more stable, the immune response is better, and that's why phages can be used as vectors to deliver drugs to the certain points, to deliver vaccines. Also, a very important field, gene therapy. Thanks to the study of the bacterial immunity towards phages, now we have the technology to change the genome. For example, if we're talking about genetic diseases, we can cure them now, because previously they were like a doom. A person who has genetic disease is like the way of life now. But today it can be changed, because wrong genes can be changed to the right genes with the help of gene therapy, and this gene therapy can be performed with the use of bacteriophages. Phage display systems, biosensors, biocontrol. Phage display systems have to deal with the repeated protein subunits of the phage capsid. Remember, we talked about these proteins that build the phage. So this phage genome is, this phage display, is actually based on this principle of multiple repeated copies of the one and the same protein. And these repeated proteins are very important to the creation of biosensors, because the biosensor requires a lot of copies of one and the same molecules. And phages give the ability to receive many copies of one and the same protein. It can be used in biosensors. And last but not the least, food preservation. Some products like meat, milk can be treated with phages. The bacteria that cause the spoiling of these foods will be destroyed, and it will prolong the freshness, the life of these products. So it will keep them fresh for a longer time without using of any chemicals, any antibiotics, just a natural way to preserve food. Agriculture diseases. Phages are used to cure plants. An example is Erwinia millavora, the bacterium that causes the disease of apple trees, can be treated, can be killed with bacteriophages, and so these plants that are sick with this bacterium, they can be cured. Absolutely in a natural way, ecological way. It's now very popular eco way. So this is the eco way. Water decontamination. Well, you all know that if you drink water that contains bacteria, you will get sick. So it's one of the possible ways to clean the water from these bacteria is to treat it with bacteriophages. I'm not saying that this is the perfect way. I'm not saying that this will be the perfect water. But if we choose whether to drink the dirty water with bacteria or the water treated with phages without bacteria, I believe that people will choose to drink water without bacteria. And that's how phages can be used to decontaminate it. Corrosion control. Yes, bacteria can cause corrosion and spoiling of metals, and the treatment of these materials with bacteriophages can also keep them safe and stop them from ruining with these bacteria. So phages can also help to fight corrosion. And I'm not saying that these are all the perfect ways. They don't have negative sides. They do have negative sides. For example, phages, they provide big amounts of proteins. And these proteins can cause allergic reactions in some humans. But it's better to have an answer and to make it better rather than do not have the answer at all, like with antibacterial therapy, if we're talking about multi-resistant bacteria. So phages are probably the only way we can find to keep on fighting infectious diseases caused by bacteria. So I believe that it's enough for today. On our upcoming meetings, I hope to discuss all the topics that just previously I gave you the the idea of. And as for today, thank you for your attention and see you later. Goodbye.